would like to thank the organizers for giving me this opportunity to speak at this conference. So I'm a research fellow from the Structural Bioinformatics section at the Vaccine Research Center at the NIH. And today I'm going to talk about applying neutralization fingerprinting in antibody epitope prediction and predicting antibody specificity HIV-1 sera. So just to give a quick overview of the Structural Bioinformatics section, our mission is to develop computational tools and then apply them in structural analysis, sera analysis, and designing antibody and immunogen. And these all relates to the main mission of the Vaccine Research Center, which is the development of vaccines with a heavy focus on HIV-1. So Dr. Peter Guang is the PI of our section, and one of the staff scientists, Dr. Evelyn Georgiev, also plays an important leadership role in the research I'm going to discuss today. So as Dr. Wyatt mentioned earlier in his talk, during the past decade, there are a lot of neutralization antibodies isolated from patient sera, and they target on a couple of specific sites on the HIV-1 trimeric spikes. This includes the CD4 binding sites where VRC01 and NIH45, 46 binds, the V1, V2 region where antibodies such as PG9, PG16 binds, and there's also the glycan V3 region, and also the EMPER region near the viral membrane. And during the past year alone, there are a couple of new epitopes are identified. This includes the epitope of 35022, PGT151, and ANC195. And they are all located at the junction between GP190 and GP41. Okay, so in today's talk, I'm going to talk about two methods which could further facilitates the isolation of new HIV-1 neutralization antibodies, and also to assist in HIV-1 vaccine design. So the first method I'm going to talk today is a fast and accurate method to predict HIV-1 antibody epitope in residue level. So with the knowledge of antibody epitope residue information, one can further understand the resistant mutation for this antibody, facilitates the optimization of neutralization antibodies, and provides a basis for epitope-based vaccine design. The second method I'm going to talk today is a fast and accurate method to delineate HIV-1 sera specificity. With this tool, this can be used to isolate new monoclonal antibodies that may be of therapeutic value, and then it could also be used in elucidating immunodominant epitopes in patient sera. Okay, so first part of the talk. So there are currently a number of methods exist for predicting epitope residues. This includes a number of experimental methods such as crystallography, MMR, cryo-EM, alanine scanning, and PET scan. There are a couple of additional computational methods which could perform this task as well, including protein docking and analyzing sequence and structures. So in general, the experimental method has a much higher accuracy than the computational method. But the problem with experimental method, it takes weeks or months to conduct. And as computational biologists, we want to develop a method which has very high efficiency, but has a much higher prediction accuracy than the current computational method. So the method we come up with for predicting antibody epitope residue is to analyze the covariation between neutralization potency and sequence. Analyze the covariation between neutralization potency and antigen sequence for diverse viruses. So when you perform when we want to assess how good the antibody is, we usually perform the neutralization of the antibodies on different viral strains. So when you have that experimental data, you can actually first align the antigen sequence that was tested in the neutralization panel, and then align them in association with the neutralization potency of the antibody to live virus. From this, you can see that at each amino acid position, in this case, it's a GP140, you can see that at each position there is an amino acid fingerprint, which is composed of different amino acid type, and with each antibody, you also have a neutralization fingerprint associated with it. So the basis of our algorithm is that when you modify the amino acid residue type at epitope position, it is more likely to affect the the neutralization potency comparing to positions which is not at the epitope. So for positions where you have a high covariation between your amino acid type and neutralization potency, it is more likely to be epitope residue comparing to residues where you have a less covariation between amino acid type and neutralization potency. So the basis of our algorithm is the following. We use the mutual information, which is a mathematical term to quantify the covariation between different variables, to calculate mutual information between neutralization potency of the antibody and the amino acid fingerprint at each residue position. And then we rank each antigen residue based on the score. So the higher the score, it means that it is more likely to be epitope residue. So in addition to the mutual information score, we also added a couple of additional information. So first is the antigen structure information. So one may ask, so when you have this antigen and antibody structure together, you will know the epitope residue. But most often time, we don't have the complex structure. But usually we have the antigen structures to start with us. And for HIV, we do so. We have a lot abundant of GP120 structure, and now with a couple of trimer structure. So we can first use this antigen structure to remove the non-surface accessible residues, as they are very unlikely to be epitope residues. And second of all, since that the epitope residues are usually located in proximal to each other, so we can give a residue which have more high-scoring neighbor residues, a higher score. And this is an effective way to eliminate isolated false positives. And finally, we have added an epitope propensity score, which is the likelihood of an amino acid type being an epitope residues. So these elements compose of our final scoring function. So to evaluate our algorithm, we first tested our algorithm on 90 HIV-1 antibodies, where neutralization information of 180 viruses and also their antigen-antibody complex crystal structure are available. So from the antibody complex structure, the true epitope residue is defined, which is what we use to evaluate our prediction. And here you can see a list of antibodies, including a number of CD4 binding site antibody, V1, V2 antibodies, Emperor antibodies, and CD4I antibody. So this is our prediction. So with using the mutual information of neutralization and amino acid fingerprint alone, you can see that we are able to get a prediction accuracy of true positive rate at of an average true positive rate at 0.2 at the FP rate of 0.05, so which is a four-fold improvement from random selection. With the addition of structure and propensity score, we are able further to push the prediction accuracy to a TP rate of 0.4 at a false positive rate of 0.05, which is a fold increase from random selection. So for more detail, you can look at our publication. So from the previous slide, you can see that the prediction accuracy is pretty good, but now we want to see if we could prospectively predict an antibody epitope, which is not where the epitope residues was not known beforehand. So here I'm going to show you a case study where we predict the epitope of HIV-1 antibody ANC195. So this antibody was isolated in Michelle Nussensweig's lab using a stabilized HIV-1 GP120 core, which is called 2cc. So this GP120 core is used to isolate CD4 binding site antibody, but however, this antibody ANC195 doesn't possess any characteristics of CD4 binding site or CD4 antibodies. And at the time of prediction, we don't have the ANC195 GP120 complex structure. And we don't really know any experimental data about this antibody in terms of their epitope. So this slide shows a prediction of our algorithm. You can see that the top 10 predicted residues from our algorithm, they are not located in the CD4 binding site, which corresponds to the previous experimental evidence. And it is interesting to see that 3 of the top 10 predicted residues are part of 2 unlinked glycosylation sequence at GP120 residue 234 and 276. So it suggests that this antibody might actually bind to these 2 glycans. So we went ahead and validated our assumption. So we first tested the ANC 195 binding with a couple of GP120 mutants where we knock out either one of the two glycan sites that we predicted. And from the ELISA, you can see that this antibodies bind to the wild type, but when either one of the glycan was knocked out, there was no binding at all. And similarly, we want to see if this effect is also present in the neutralization experiment. So we knock out these glycans from the pseudovirus that we tested for neutralization. And again, when we knock out either one of the two glycan sites, the ANC 195 neutralization was completely eliminated. So our prediction was further validated with the recent publication of the ANC 195 GP120 complex structure. As you can see on the right, this is the new published actual crystal structure. And you can see that the epitope indeed includes glycan 234 and 276. And 5 of our top 10 predicted residues are part of the true epitope ANC 195 determined by the crystal structure. So this is a pretty good prediction accuracy. So we also have implemented a web-based server called NEP, which stands for Neutralization-Based Epitope Prediction. So this is available to the public, that people can use this algorithm, this website to run our algorithm. So the website requires 2 input. So you need to have an antibody neutralization data of diverse virus sequences. And you also need to have the antigen sequence of the viruses that you tested. And you can also supplement antigen structure, if you have it, to enhance prediction accuracy. So to conclude this part, we have developed a neutralization-based algorithm to predict epitope residue of HIV-1 antibodies with good accuracy and high efficiency. And we have predicted the binding mode of ANC 195 before the crystal structure was published. And we also have implemented a web-based server, which is available to the public to run our algorithm. And finally, I show you the applicability of our algorithm for determining HIV-1 antibody epitope. Theoretically, this method could be applied to other viruses as long as there is diversity in it. For example, this should be very applicable to viruses such as influenza. Okay, the second half of my talk in predicting HIV-1 sera specificity. And again, there's a couple of experimental methods which could already do the work here, including peptide interference, epitope-specific probes such as GP120 core, knocking out specific sites on probes or viruses, and also generating chimera viruses. So all of these, again, takes weeks and months to run, to conduct, and we want to develop something which could be where we can get the results in a couple of seconds or minutes. So again, our method is based on analyzing neutralization fingerprints. So as I mentioned previously, with each antibody, you have a unique neutralization fingerprint. And different types of, for antibodies targeting similar epitopes, they should have similar neutralization fingerprint. So now, if you have the neutralization fingerprint of the serum, you can actually deconvolute the neutralization fingerprint of the serum with the linear combination of the neutralization fingerprints of antibody representatives of different major epitopes. So you can, from this deconvolution, you can see that which types of antibody present is dominating the neutralization signal of the serum. So here are a couple examples. When we applied our protocol to donor 45 serum, which is a famous donor that there are a number of VRC01-like antibodies were isolated from. Indeed, you can see that the product predicts that VRC01 has the highest contribution to the sera signal. And similarly, for N152, for analyzing the N152 sera, we were able to predict that the 10A-like antibody, which targets the C-terminal part of the emperor region, as the highest contributor. And indeed, this is a serum where 10E8 antibody was isolated. We have also applied our algorithm to detect the serum specificity for CAP2 for patient CAP256, with which we have the sera in different time points. And as you can see that from as the disease progress, you can see that the signal for V1, V2 for PG9-like antibody, which targets V1, V2, the signal actually increases. And later we were able to isolate a new antibody called VRC26, which is indeed is very similar to PG9 in the sense that it targets also the V1, V2 region of the amyloid spike. And we also want to see if our computational analysis can have any correlation with the standard experimental mapping methods, such as using GP120 core and other knockout probes or viruses. So we perform a study of applying our prediction and standard experimental methods on 21 different patients' sera. And as a result, you can see that more than 85% of the cases, one of the top two specificity that the computational algorithm picked up, it was validated as a true positive by the experimental mapping methods. Suggesting there is some degree of correlation between the computational method and the experimental mapping methods. Okay, so to conclude this section, we have developed a new computational tool based on neutralization fingerprint to predict HIV-1 sera specificity. And this tool can aid the identification of new HIV-1 broadly neutralizing antibody and understand the development of broadly neutralizing antibody during the course of infection. So here I show you the changes of specificity in just one patient, but then if we have an abundant sera of sera with multiple time points, hypothetically, we can also use this tool to look at the immunodominant epitopes in a population level. And so finally, for acknowledgement, I would like to thank Drs. Peter Chuang and Evelyn Georgiev for their leadership in this study, and members in structural biology section for providing the structures and also for doing some biochemical analysis, and members from Dr. John Mascala's lab for performing neutralization experiments, and David Liu for implementing the web server. And thank you very much for your attention.
Let's take a couple of questions. Yes.
Can your approach be used for purely binding? Does it have to be neutralization?
Yes. Actually, this is a very good point. It doesn't have to be neutralization. If you have this binding data to targeting diverse viruses, this should work as well.
Why?
Because it is, actually, let's go back to it first.
Binding is just sort of binding. Neutralization is actually doing something.
Yes, but ideally neutralization is better, but if you have binding data, you can still, basically you can see how each residue position has an effect. Epitope position affects binding, right? So if you alter the epitope positions, it will affect. Actually, binding probably makes more sense in this case than neutralization. It's just that we have a neutralization data in handing, or we usually don't do a panel of binding analysis. We have neutralization data, but binding is epitope is a place where antibody binds. So theoretically, if you have binding data, it actually has a more direct effect than neutralization.
I wouldn't agree.
Why?
Because, you've got a lot of stuff which sort of says antibody-antigen interactions happen. Okay, fine. Yeah. And they may lead to precipitation. Fine. But when you have a viral neutralization, you have a physiological effect on the virus, which actually means something to protection. Binding is just cosmetic. It just looks this way or it looks another way. So I would, if I were involved in this project, I would say that binding is a total utter waste of time. What you're really looking at is virus neutralization and what are the epitopes that are involved when associated with an antibody in neutralizing the infectivity or the physiology of the virus. I'm not interested in the cosmetics of the virus as what it looks like when it's got an antibody attached to it. It's totally irrelevant, surely.
I'd like to suggest that maybe Dr. Spear,
Okay,
you can talk to each other. And
sure.
Let's have a vote.
Yes,
Oh, we can do a vote as well.
join us late.
Yeah.
We lost 20 minutes in the beginning, therefore we have to take a 5-minute break and come back, and then we'll do the next.
Can I apologize? I didn't realize that I was involved in this afternoon's session. Otherwise, believe me, I would...
Helen's supposed to be chair.
Yeah, but he was supposed to be chair this morning too. But he told me to basically take over for the morning. I appreciate you were waiting for me, but I didn't realize that it went on all afternoon as well. So forgive me for that, and it was completely misunderstood. It was a binding business, not a neutralization business.